Hello friends and my dear students, myself Komal Valde from RBNB College, Srirampur, district Ahmednagar. Students, today we are discussed about the biology practical of standard 12th. From biology practical, we have to discuss about the question 4th, that is spotting. Student, there is a question 4 of spotting and there is the 10 spotting from A to J. And today we are going to discuss about the spot D, that is the permanent slide of the TS of testis, TS of ovary and VS of blastula. So students, the structure of that permanent slide is easily observed under the microscope and you have to write on comments on these 3 permanent slide or one of them. So we start now the first slide, that is the TS of testis. It is the TS of testis which consists the seminiferous tubule and tunica albuginea. The TS of testis is lined by the tunica albuginea. Inner to the tunica albuginea there is presence of the tunica vascularis and it is externally covered by the tunica vaginalis. There is the 3 to 4 seminiferous tubule presents under the presence in each locule. That seminiferous tubule consists the inner lining, that is the germinal epithelium. This germinal epithelium there is the presence of pyramidal shaped cells. These pyramidal shaped cells are called as the Sertoli cell. In the seminiferous tubule there is the gametogenesis, means the process of spermatogenesis takes place, means from the germinal epithelium there is the development of sperm or spermatozoa takes place in that seminiferous tubule. There is the inner lining of the germinal epithelium. Then there is the primary spermatocyte and secondary spermatocyte, which is the phase of spermatogenesis, and this primary spermatocyte is formed the spermatozoa or the sperm. The function of that Sertoli cell which is present in between the germinal epithelium, this Sertoli cell provide nutrition and nourishment to the developing sperm. In between the seminiferous tubule there is a group of cell. There is the group of cell which is called as the Leydig cell or interstitial cell. That Leydig cell or interstitial cell is produce the male hormone androgen, which is testosterone. It is the structure of TS of testis, and you have and students you have to write on the comment on that TS of testis. And after this observation of slide you have easily write on the comment of that slide. Now we are moving our second slide, that is the TS of ovary. It is the second slide of the spot D, that is the TS of ovary. In the TS of ovary it is divided into 2 regions, that is the ovarian cortex and middle medulla. The ovarian medulla and there is the cortex is present. In the cortex of ovary it is again lined by the germinal epithelium. This lining of the ovary is called as the germinal epithelium, and in that cortex region there is the oogenesis takes place, means from the primordial follicle there is the development of Graafian follicle. This oogenesis process takes place in the cortical region of the ovary. There is the primordial follicle. After that the primordial follicle it develops into primary follicle, secondary follicles, and lastly it is developed into the mature follicle. That mature follicle is called as the Graafian follicle, and this secondary follicle is produce the female hormone estrogen. This Graafian follicle or mature follicle consists the follicle outermost layer is the follicle, and inside this there is the presence of ovum. This ovum is firstly known as the oocyte and then it is developed into the matured ovum and it is present in the mature follicle or Graafian follicle. After the 14th day of the menstrual cycle, this Graafian follicle is ruptured and this ovum comes out from that Graafian follicle, and this process is called as the ovulation. This process is called as the ovulation. This ovum comes out from that ruptured follicle, and this ruptured mature follicle is turns into the corpus luteum. This corpus luteum produces the hormone progesterone if the ovum is fertilized with the sperm. If the ovum is not fertilized with the ovum, this corpus luteum yellow body is turns into the white body, and this white body is called as the corpus albicans. Student, you have to write on the TS of ovary when you observe that TS of ovaries or the slide of that TS of ovary, and you have to write the comments on that TS of ovary. Now we are moving our third slide of the spot D, that is the VS of blastula. So student, if you observe this slide under the microscope, you have to see this structure of the VS of blastula. This blastula is the stage of embryonic development. This blastula is the stage of embryonic development, and here the outermost layer or outermost cell layer is present in the blastula. This cell of the blastula is called as the trophoblast, and this trophoblast contain a cavity in between the trophoblast. This cavity is called as the blastocyst cavity, or it is also known as the blastocele. At the one end of that trophoblast, there is the group of cell is present attached to the trophoblast. That group of cell attached to the trophoblast, these cells are called as the cells of Robber, and inner to the cells of Robber there is the inner cell mass, which is called as the embryonic knob. This inner cell mass is called as the embryonic knob, and it is attached to the one end of the trophoblast, and that cell is called as the cells of Robber, and from that there is the embryonic development is consist. And student, these three slides, namely the TS of testis, TS of ovary and the VS of blastula, are the spot D of the question 4th spotting. Thank you.